Anyone know what day of the week this is? Thank you. Thank goodness. Hi, my name is Bernard Siegel. Welcome to the Friday. It is Friday, right? December 9th. And this is the fourth day of the 12th Annual World Stem Cell Summit. I'm Bernard Siegel, Executive Director of the Regenerative Medicine Foundation and the founder and co-chair of the annual World Stem Cell Summit and RegMed Capital Conference. For those of you that are watching the live stream, thank you for this. We're archiving, of course, and we'll be showing all of the sessions, certainly from the Grand Ballroom, available to you, and other sessions that are being recorded as well throughout the meeting, and all of those that have been looking at the AMP2 interviews, and we've gotten a tremendous social media response for that, really enormous. We appreciate all the kind words and retweets and things that have happened. Well, this is the last day, but those of you that canceled your flights and extended your stay in warm South Florida at the Palm Beach County Convention Center will surely be happy, because this is the day when we can all breathe a little from all the chaos, all the receptions, and everything else that's gone on. We can enjoy our last hours at the exhibit hall during a break this morning and say goodbye to our exhibitors and exchange business cards. But also it's an opportunity to reflect and have more dialogue on some very interesting sessions. And I'm very pleased that this morning we have two plenary talks that I am personally very eager to listen to myself. We have first a talk provided by Martin McGlynn, who happens to be a member of the board of directors of our organization, the Regenerative Medicine Foundation, but has been involved with industry and ran the company Stem Cells Inc. for 16 years before he retired. There's no one that understands the challenges of creating a cell therapy product that I know who's better versed in the industry side of this and the challenges than Martin. And before I bring him on the stage, I just wanted to let you know, following his address will be a talk by Dr. Ilyas Sinjek. Now, I'm really happy that Ilyas has joined us today. He's the head of stem cell research at the National Institute of Health Center for Regenerative Medicine. And having NIH to be able to present what they're doing here and now related to our field is vitally important, so we're very honored. And I'll bring him up in about a half hour. So let me bring up to the podium Martin McGlynn. Please give him a warm round of applause.
Thanks, Bernie. Thank you. Good morning, everybody. Thank you for getting up out of bed on the final day of the conference. Gold medals all around. I appreciate you being here, and those of you who are live streaming, welcome as well. Today I want to give you kind of a 50,000 feet in the air summary, personal perspective, some of my own experiences over the years, engaged in translation of pioneering cell therapy. It's a very personal overview. It's a variation in some ways on a theme that started to talk about on Wednesday morning, the plenary session with Dr. Whitten. Every now and again I go back and I want to re-educate myself in terms of what I think of in the context of translating cell therapy and translating scientific discoveries to turn great science into groundbreaking medicines. And the one I use is this one, which really the focus here is when you're using the cell as the therapeutic agent, as opposed to the cell being the target in the body, using other modalities. And so the look back that I'm going to provide you today is essentially going to be focused solely on unmodified cells. I thought it would be worthwhile taking a look at the arc, if you will, bringing us up to today. in the history of unmodified cells. And of course, you have to go back to the 60s to McCulloch and Till, two great Canadian scientists. The first major industrialization effort to bring cell biology into an industrialized environment was with after the discovery of the mouse hematopoietic stem cell, Irv Weizman's lab in Stanford, creation of a company called Systemics, where they discovered the human version of that cell, their acquisition by Sandoz, now Novartis, and the major contribution that endeavor provided for our industry. Then you move into the 90s and you're looking at the results of great work by Dr. Arnie Kaplan, who was here yesterday and the day before. His work with mesenchymal cells. He prefers not to call them stem cells, and I can understand why, and I agree with him. And then the human embryonic stem cell discovery and the work done by Jamie Thompson led to the creation of a company called Geron, which I'm sure we all know. And then a company called ACT was established to develop embryonic stem cells derived RPE cells. And that, of course, involved Dr. Lanza's work. ACT changed its name along the way to Okada, and then Okada was acquired by a Japanese company, Astellas, and I'll talk some more about that later. And then into the 2000s, the human neural stem cell that was isolated and discovered by Dr. Yoshida and her colleagues at Stem Cells Inc., first published in PNAS in January of 2000. Work done by Carl Joe at the NIH that led to the formation. We co-founded a company called Neuralstem, and then another company called Reneuron, which is a UK company, and Dr. John Sinden was one of the co-founders of that company. And then work done by Catherine Verfay led to the formation, led to the expansion really of the horizon and focus of a company called Athersys, which has been involved over the years in the development of multipotent adult progenitor cells. And more about that in a moment. So you're looking here at about a half a century of history in the work of these great rock stars who brought this cell biology to the industrial world, if you will, and presented people like me and my colleagues with some very significant challenges and great excitement. There were many trials and tribulations along the way with these legacy platforms. The hematopoietic stem cell technology ultimately was abandoned by Sandoz when it became apparent that the technology would not lend itself to ex vivo expansion, and a patient-specific business model for a pharmaceutical company was not all that attractive. That said, I read recently that more than 1 million patients have received hematopoietic stem cell transplants to date and have greatly benefited from them. Athersys partnered with Pfizer. They conducted a phase 2 trial in inflammatory bowel disease. They did not meet the efficacy endpoints, and Pfizer turned back the rights. Athersys refocused and is now pursuing, among other things, ischemic stroke in the United States and in Japan with their new partner, Helios. And Helios partnered with Athersys after Chugai, who had originally licensed the rights, returned those rights to Athersys. So a little bit of a bumpy ride there. And then Osiris sold its culture-expanded mesenchymal stem cell assets to Mesoblast in 2013 for approximately $50 million and another $50 million in other consideration. Mesoblast is in phase 2 for heart failure, rheumatoid arthritis, lower back pain is in phase 3, and they've actually got product approved for graft versus host disease in Japan. Interestingly enough, Teva had partnered earlier on with Mesoblast, and they returned the heart failure rights to them. We all know, of course, about the tortuous journey of the embryonic stem cell technology, which predominantly was stymied by safety concerns about tumorigenicity, complex differentiation protocols to terminally differentiate the primitive embryonic stem cells into transplantable cells, and then, of course, significant political and religious objections, not just in the United States. Geron licensed the drug screening rights to this platform to GE Healthcare many years ago, and then ultimately sold their assets to Biotime in 2013 for $5 million in cash and other considerations. I'm pleased to say that Asterias, which is a subsidiary of Biotime, is engaged in the conduct of a clinical trial in cervical spinal cord injury. Again, this is a very early study. Okada, which I mentioned, previously known as ACT, was sold to Astellas in 2016. And in the case of the technology that we were developing at Stem Cells Inc., we were stymied from the get-go by risk-averse regulations. Fetal tissue, which was the source of the cells, which is the source of the cells, was also controversial. The use of immunosuppression, because this was an allogeneic procedure, was also a factor that had to be taken into account, not just by the clinicians, but also by the patients. Not so much a big issue in normal, healthy adults, but certainly a limiter in the conduct of clinical trials in more elderly patients. Interesting, the early trials in the pediatric population tolerated immunosuppression extremely well. Stem Cells Inc. And Neurostem exited this year. I'll talk some more about that later. Reneuron in the UK is still standing and in the fight. And they have a significant strategic investor in the UK, and I think that's one of the major factors for their continuation and hopefully their success. So I'd like to share with you some observations that and these are kind of random in a way, but you know, we've heard all along in this conference how long it takes to develop medicines. It is important that we do great science. It is important that we be regulated. It is important that we design robust clinical trials and we ask the tough questions. But unfortunately, the runways are way too long and they're way too costly. In the case of Stem Cells Inc., we invested somewhere in the region of $300 million getting to early phase 2 studies. So that's a significant amount of money over a period of 15 years. And of course, investor fatigue sets in. When you look at the companies engaged in translation in the cell therapy arena, they typically have small market cap valuations. And I'm not going to go into all the technical rules of Wall Street, but your market cap has a great dampening effect on your ability to raise significant amounts of capital, as a result of which companies are forced into serial punitive financings. There's, in turn, significant dilution, which investors do not like, and you get into a vicious circle because the investors don't like it, and then the market caps start to shrink. And as that happens, and your stock price goes under pressure, then you have to deal with the listing agencies, whether it's NASDAQ or New York Stock Exchange, and you have to meet certain listing requirements as a public company. So in order to avoid delisting threats, companies are many times forced into reverse stock splits. What's also frustrating is that when you're really in the pioneering space, you're the tip of the spear, and you get to a point where you can actually get the green light to do a phase 1 safety trial, and you're looking for signals of efficacy. The phase 1/2 open label safety and efficacy data, which for the most part has been very exciting, was not valued by investors. You could speculate that maybe the industry created its own problem by overly hyping what the expectations might be for the technology, and when the hype wasn't met, the investors yawned. But that's up for discussion and debate. When phase 2 controlled studies, those limited number of studies did come onto the radar, they didn't excite investors that much, quite frankly. And of course, the pharmaceutical industry and strategic investors, or the smart money, somebody said the smart people, they sat on the sidelines because they wanted to see robust phase 2 data. So then you're back into the cycle again, that funding for additional trials, because of investor fatigue, low market caps, the funding dries up, and when you do a funding, it's overly punitive for the existing investors. And so what happens then is, nature hates a vacuum. New technologies fill that vacuum. And we have exciting technologies on the radar screen today, whether it's iPSC platforms or great tools like CRISPR-Cas9 gene editing capabilities, immunotherapy, immuno-oncology, and on one could go. So the way I like to describe it is that we're really still in the beginning of harnessing the full potential of cell-based medicine. Some learnings with regards to the legacy platforms. Cell source matters. It matters not just scientifically, but it also matters in terms of acceptance, regulation, political and scientific, political and social interest or lack thereof. And as you know, we have a whole plethora of sources, whether it's embryonic, fetal, placental, bone marrow, peripheral blood, adipose tissue, et cetera, et cetera. We could go on. I thought about putting in bagels from Brooklyn as I was going through the list. I've looked, there aren't any cells in Brooklyn bagels. That's a little side joke. The US ban on federal funding for embryonic stem cells in 2001, and subsequently the European Parliament's ban on the destruction of human embryos, cast a shadow over the entire cell therapy field, not just the embryonic stem cells. Because the general world at large were not able to differentiate between an embryonic stem cell, a tissue-derived cell. So it's the lineless effect, it's the dark shadow that you have to continuously walk around under. And I don't think the legacy platform companies, the original pioneers, really were able to get out from underneath that. Immunosuppression, as I've talked about, is a barrier to scale up and to widespread adoption of allogeneic therapies. And a lot of work needs to be done in that field of endeavor. Some great theories out there as to how one might go about doing this, including induction of immune tolerance and various other gee-whiz approaches now to immunosuppression. And I'm sure they will all come to play in time. Robust phase 2 data is essential to attract the pharmaceutical industry and strategic investors. When you look at the autologous platform, one would think, that that's kind of Mother Nature and that's going to readily be adopted. However, the logistical challenges that are associated with autologous therapy, let alone just the cost equation, in my opinion, that's a great barrier to widespread adoption, and many others agree, and there are a lot of people who are focusing on that and bringing assets to bear. It's interesting, I was listening to a talk by the CEO of Helios recently when he talked about the first autologous procedure that was done in Japan in age-related macular degeneration, and he mentioned the fact that it cost $1 million for the cells for that patient. And so I think that speaks for itself. But in terms of scale-up for the autologous cell therapy platform, there has to be significant investment in infrastructure, in software, in logistical capabilities, et cetera, to fully leverage that platform. Another learning is a learning that I took to heart, and that is you should only do clinical trials that's based on science, based on great preclinical data in the patient population most likely to benefit. Oftentimes, sponsors find themselves in negotiation with various regulatory agencies and essentially compromising, losing sight of the fact that the patient population that they're going to put the cells into in the first instance is really not the patient population that they might likely benefit from. Rather, they fit nicely into a regulatory paradigm of risk versus benefit. And another one is that be prepared to conduct clinical trials outside the United States if necessary. But if you do that, make sure you do it in compliance with the harmonized regulations so you can bring the data back. Just one example, we did that in spinal cord injury. We could not convince the agencies in the United States or Canada to allow us transplant cells directly into the spinal cord in the patient population that we thought would benefit most from. We were being restricted into thoracic spinal cord injury in the worst of the worst patients, patients who had complete motor injury. And we just felt that that wasn't right for the technology. Bottom line, we went to other jurisdictions, and finally we found a regulator in Swissmedic who agreed with us. And so they allowed us to do an adaptive clinical trial design where we did a small number of Asia A patients and quickly moved into the Bs and Cs. We subsequently brought that data back to the United States and Canada, and we received the green light to do phase 2 clinical proof of concept studies in the United States and Canada, having shown the human data from the studies that were done in Switzerland. Putting on crystal ball here, a little bit of a look forward. Will the next generation fare better? We've got HLA-matched iPSCs, induced pluripotent stem cells. It certainly solves the cell source issue. It's non-controversial, but still requires some degree of immunosuppression, and it faces a long road through the clinic. That said, I think Japan could be a real game changer because they've upset the apple cart and they've changed the rules of the game. Key players in the field now, as a result of all of this, are companies like Fujifilm, Sumitomo, JCR Pharma, Astellas, Helios, just to name some of them, and there will be more jumping in. Hematopoietic stem cells that are HLA matched or not, used to reboot the immune system in leukemia and lymphoma treatment and potentially autoimmune disorders. Very exciting to see that technology still very much relevant. Critical success factors, though, include non-toxic conditioning regimens, improved bone marrow harvesting and peripheral blood techniques, and the cell expansion process. A couple of key players not meant to be completely inclusive are Magenta and NOLA. They're relatively newcomers in the field, but there's a lot of interest in funding the use of this technology now with new techniques and learnings from the first wave of hematopoietic stem cells. So immunotherapy for liquid and solid tumor cancers using the patient's own activated T cells against cancer, it certainly solves the immunosuppression issue, solves a whole bunch of other issues, but it adds another step into the process and another step to be regulated. And that's the gene insertion step and the chemotherapy conditioning regimens. You all know the names of these companies, Juno, Kite, Bluebird, and of course Novartis is in there very much neck and neck with Kite. Developing concerns with that technology are recent patient deaths in Juno's CAR-T leukemia trials. They're trying to work their way through that, and it's interesting to see how the market is reacting to that, how the regulators are reacting to that, and how the companies are reacting to that. Certainly very different from the first stages of gene therapy all the way back to that very unfortunate outcome in the early gene therapy trials with Jesse Gelsinger. It was a completely different reaction, thankfully. It could have had the potential to really set this whole field and this whole platform back many, many years. As I mentioned, autologous cell therapy resolves the immunosuppression issue. It needs significant investment in infrastructure due to the complex logistical challenges from patient all the way through to processing, all the way back to the patient. And that investment is essential if this is to be, this platform, very promising platform, is to be scaled up. Key players here are Quintiles IMS, recently received a CERM grant for their work to help create tools and capabilities in the early stages of translation. GE Healthcare and GE Ventures-backed initiative with Vitruvian Networks, who are essentially focusing on the connective tissue and all of the logistical challenges associated with patient through processing back to patient. And then we have universal cells, where the goal is to produce non-immunogenic cells, pluripotent master cell banks that can be used in an allogeneic way, on demand, if you will. Very, very early technology. And also, you could talk about organ buds and the technology involved in that technology, which is very exciting if it works. So I think the answer to the question is, will they fare better, is most likely. We've spent a lot of time at this conference talking about the Cures Act, which is very welcome. It certainly provides more tools to the FDA. It does, however, leave a lot of discretion in the hands of the FDA. And in my own opinion, I think that's good. Because with this technology, one size does not fit all, and each sponsor has to figure out with the regulators what is the best pathway. They say they're open to talk. They say they're open for business. They want to help this technology advance quickly. Take them at their word. The old Eastern motto of to define is to confine. And I think in the context of the Cures Act, the fact that there is a lot of discretion left into the hands of the FDA is really good news. Has to be worked. In November of 2013, as I'm sure you all know, Japan established an expedited pathway to approval for cell therapy products. And this came on the heels of Yamanaka's work with iPSC platforms, reprogramming. As you know, he received the Nobel Prize in 2012, and Japan stepped up to the plate. They looked at this technology as strategic, and they decided that they were going to go full bore, enable the technology, and do some other things, which I'll mention in a moment. But clearly, it was a bold and disruptive move. It's already accelerating cell therapy treatments and cures. It creates a competitive advantage for Japanese companies with this accelerated pathway. The rules are pretty clear, unequivocal. It creates incentives also for foreign companies to partner with the Japanese companies. And crucially, this move by the government in Japan has successfully lured the major pharmaceutical companies into the field, something that was lacking with the legacy platforms. Japan is on the move. In 2014, Dai Nippon Sumitomo formed a joint venture with Riken and Helios to develop IPS technology focused on disorders of the eye, the back of the eye. In March of 2015, a company formed originally by Jamie Thompson, Cellular Dynamics International, was acquired by Fujifilm for a little over $300 million. In November of 2015, as I mentioned earlier, Estella acquired Okada for almost $400 million. And in February, Mesoblast had formed previously a joint venture with JCR Pharma in Japan, and they gained full approval in Japan for graft versus host disease. They're now seeking conditional approval for heart disease, and it really is the camel's nose under the tent, and they're going full steam. In April of this year, Universal Cells, a new company, formed a partnership with Helios. So you see Helios is being very active in this field. And in September of this year, they got a green light, a trial approved for an approval trial for stroke by the PMDA, despite the fact that they had some disappointing phase 2 results in their ischemic stroke trial. They've repositioned that, redesigned the protocol, and they're off to the races again in Japan, which is encouraging. China is not far behind. The moratorium on clinical trials in China is still in place, to my knowledge. But there are guidance documents that are slowly and carefully emerging. Based on my own information and own experience and interaction with various stakeholders in China, they definitely intend to be competitive with Japan. And so they're taking their time and making sure that their regulations will be able to compete on the global stage. China is investing in science. Companies are twinning with academic research institutions. They're physically locating proximal to the institutions, and they have essentially, in some cases, physical bridges, physical walkways to these universities. And there are guaranteed jobs, if you will, when the newly minted scientists come out of academia, which is tremendous. Chinese companies are acquiring assets and know-how, and it's the know-how I personally think is invaluable. A Chinese company called BioLife took an interest in Seska, stepped up their holdings in Seska, and now have gained control of. It's a California company. Used to be known as Cool on NASDAQ, KOOL. It is a cool company, and they've got cool technology. You should check them out. And then Bright Oceans Corporation of China recently acquired all of the cell assets, the neurostem cell assets, IP, data, know-how, cell banks, and they are doing extremely well. It looks like they intend to continue to advance this technology. They're taking a very strategic view, the longer view, of the potential value of this technology for disorders of the central nervous system. So in summary, we definitely need a more flexible regulatory framework that reflects the unique challenges associated with cell therapy, and we need to balance the need for sound science with accelerated development pathways. We can't compromise on either, in my opinion. New tools have been given to the FDA. It's a step in the right direction. Breakthrough designation, expedited programs, accelerated approval, etc., for life-threatening or serious conditions, and involving patients in the dialogue. But a 10 to 15-year runway to clinical proof of principle is the valley of death, and that was talked about before. And Mike West did a great job yesterday in his talk, but you'll notice he also mentioned that. And that's when you get from, the great science to the clinical proof of principle, and you're on your own in terms of trying to find funding for that. And in that environment, the financial and human capital would go elsewhere, maybe into iPhones, maybe into laptops, who knows. I would just say this, that I think there is a great need for shared responsibility for translation of scientific discoveries between government agencies, academia, pharma and biotech, and cell therapy companies. We have to find a way to bring everybody into this game. And personally, I think that too many of our technologies and scientific breakthroughs and discoveries are coming out of academia too early. And we see signs now where academic research is staying within the academic institution, and work is being done, not necessarily owning all of the assets, but work is being done to leverage assets and know-how around the globe to get the clinical data before bringing it out and making sure that it's ready for prime time. So I want to thank you again for getting up early this morning, and appreciate you being here, and anybody has any questions, I'll be glad to take them offline. Thank you very much. All right, thank you. Take care.
Thank you, Martin, for that overview, which I thought was brilliant. I'm very pleased to introduce Ilya Sinjek. Dr. Sinjek is the head of stem cell research, NIH Center for Regenerative Medicine, National Center for Advancing Translational Sciences. This is an uncertain time, right, for NIH. We're wondering what the next step is, where the funding is going to be. And it reminds me of a quote, I think that originated with Irv Weissman, that the National Institutes of Health is the greatest engine for scientific advancement in the history of humankind. Think about the enormous investment that has come over the years. So we support NIH and appreciate everything NIH does in advancing this field. So let me introduce Dr. Sinjak.
Okay, good morning, everyone. It's a pleasure to be here. It's a great honor. Thank you so much for the kind invitation. I want to basically tell you about our program at NIH. So we started a laboratory a little over a year ago called the Stem Cell Translation Laboratory. This is housed within the National Institutes of Health, and we have also an institute or center called the National Center for Advancing Translational Sciences. And I want to basically make the point that this is the next phase of what was previously known as the NIH Center for Regenerative Medicine. Now it's actually called NIH Regenerative Medicine Program. So in today's talk, I want to touch on these three areas. So give you quick overview on iPS cells and some of the main challenges that we're dealing with after now a decade working with these cells. In the second part, I want to, basically give you more information on our laboratory at NCATS, and then also introduce you to some of the ongoing projects, ideas, and resources that we are working with. So as you all know, development starts with the fertilized egg, and so through multiple cell divisions, a blastocyst stage embryo is formed, which harvests the inner cell mass, which then can be cultured and commonly known as the embryonic stem cells. These cells then give rise to multipotent organ-specific stem cells, which are the building blocks of organs, and further down the road, these cells become more specialized and make up these different organs. So over time, there's a decrease of potential and increase of the lineage commitment process that takes place. So the first embryonic stem cell lines were established by Jamie Thompson in 1998, and as I just pointed out, these cells come from the inner cell mass. So in groundbreaking work done by Shinya Yamanaka in 2006, it was demonstrated actually that you can turn back time in cells by introducing defined transcription factors, so known as the Yamanaka factors, so OCT4, SOX2, KLF4, and SUMEC. So essentially, this is a process that does not rely on embryos, but rather taking somatic cells from any patient and generating cells, so-called induced pluripotent stem cells or iPS cells that behave in many ways similar to embryonic stem cells, and that they can differentiate in all human cell types and then hold potential for regenerative medicine and cell therapy. So to give you a brief overview on how this process works. So essentially, after introducing those factors, ordinary fibroblasts derived from the skin basically undergo morphological changes, and the genetic program is changed, and over the time course of 3 to 4 weeks, these cells become these iPS cells. And this is a very early experiment that I started doing actually back in San Diego immediately after the second paper by Dr. Yamanaka was published in 2007, that this technology actually works with human cells as well. So again, morphology-wise, differentiation potential into embryoid bodies, and giving rise to a number of different cell lineages, we can demonstrate that they fulfill the basic criteria of being pluripotent cells. This also includes teratoma formation, in this case, injecting undifferentiated cells into mouse models, and you get these tumor formation in animals that you can further study, and another step in demonstrating full pluripotency of these cells. So now using more specific differentiation protocols, we can generate very specific cell types. In this case, neurons, astrocytes, and oligodendrocytes can demonstrate long-term growth of these cells and also karyotypic normal state. Taking this even further, showing that these neurons actually are functional. So these cells generate action potentials. They are spontaneously synaptically active, having miniature iPSCs, and can respond to certain pharmacological substances. So demonstrating essentially that parental fibroblast now has been turned into a neuron and fulfills all criteria that we know of neurons should fulfill. But what are the major challenges remaining in the field? So this is really important. So after a decade now, we are at the stage where we want to move this technology closer to drug discovery and cell therapy. So first, making and using iPS cells actually in a standardized fashion. So we still see many different variations to protocols. Biological complexity is obviously a very important problem that we want to tackle. Technical variability and establishing robust SOPs. The second point is directed differentiation, actually. The cell types of interest, how can we generate these cells rapidly in a highly controlled fashion and maintain safety, which is the third point. So when going into the clinical arena, we want to make sure that we don't carry over undifferentiated cells that potentially could cause problems in the patient. So how can we control and avoid teratoma formation and carrying over undifferentiated cells? So these are the three main areas I wanted to emphasize today. And actually, Dr. Yamanaka himself is looking carefully into these questions. In these two papers published in PNAS, it's actually showing that there is marked differences in these differentiation propensity of various human iPS cells. And what are the reasons for this? So I want to highlight first that, switching gears, that the National Center for Advancing Translation Sciences was established in 2011 by NIH. So the goal here is to catalyze the generation of innovative methods and technologies that will enhance the development, testing, and implementation of diagnostics and therapeutics across a wide range of human disease and conditions. So this is the mission statement of NCATS. So as part of this center, when I joined a little over a year ago in 2015, the mandate was to actually establish now a stem cell laboratory within NCATS. And this is, again, the continuation of what was previously known, the NIH Center for Regenerative Medicine. Now it's called the NIH Regenerative Medicine Program. So we want to use cutting-edge technologies in the dedicated laboratory space over 4,400 square feet. So bringing together a multidisciplinary team that works to address these important questions in the field. And this team is composed of folks that have been, worked in industry. We have automation engineers, colleagues who know compound management, project management, toxicologists, et cetera. So really a whole team of highly dedicated scientists to work on these ideas. And the 3Ds of NCATS, so we want to develop new technologies, demonstrate the utility, and disseminate it to the public. So more specifically, the goals of the Stem Cell Translation Laboratory that I'm leading. So first of all, so establish quality standards to define pluripotency and differentiated cell types in more precision and in greater detail. So we want to understand heterogeneity of cells while they differentiate. We want to develop methods actually that help us to mature cells. This is a problem in the field. Oftentimes we end up deriving cells that express certain markers, but they're not fully mature. So how can we really push these cells to full maturity? And finally, to make this process also very practical in terms of replacing synergenic material or recombinant proteins by using small molecules, for instance. So the basic problem shown here. So after deriving iPS cells, so understanding these transient cell types in between and ending up with somatic cells that are mature, this is, I think, one of the major problems in the field. And we want to fulfill certain criteria, make this feeder-free, controlled, robust, scalable, and eventually suitable for cell therapy. And the first cell types we are actually focusing on is shown here on the right-hand side. So this includes dopamine neurons, motor neurons, serotonergic neurons, astrocytes, oligodendrocytes, hepatocytes, and insulin-producing beta cells. So this is an overview of NCATS DPI. So this stands for Division of Preclinical Innovation. We also have a Division of Clinical Innovation. I'm not going to talk about that division today. But essentially, we want to highlight that the Stem Cell Translation Laboratory is surrounded by other groups that can help us with functional genomics, like whole genome RNAi, chemical genomics, 3D bioprinting. There's lots of expertise in assay development, medicinal and analytical chemistry, etc. So this is really an effort that is not just done by one laboratory, but and this is, again, all under one roof. So one great area of expertise that NCATS has developed is quantitative high-throughput screening. So we have access to almost 500,000 small molecules. We are screening routinely over different concentrations, which helps us to reduce the number of false positive and false negatives. This is a fully automated system. We can actually screen up to 1 million wells a day. This can be miniaturized, so 15 and 36 well plates is being used routinely. And this also includes high content imaging at the same time. So we can now screen using more targeted libraries. I'm showing this to give you an idea of a new library that we established called NPACT. So essentially, small molecules, over 10,000 small molecules. So trying to cover over 7,000 known mechanisms. So we want to really use these libraries to figure out how we can make differentiation protocols more efficient. And this also includes actually in-house chemical synthesis of small molecules that we're interested in. So another area that we have leveraged is actually the use of purified natural products on iPS cells. This includes highly pure compounds, and we know their structures, and we know how to use these libraries for our screening efforts. So another area that NCATS has great expertise is actually drug repurposing. So there are two approaches to discover drugs. So one is to go the traditional road, essentially, and this takes up to 15 years or even longer. But if you now take compounds that have been in clinical trials and try to repurpose them for your other applications, you may actually save lots of time and money and actually bring these important drugs that have been tested in the clinic to the different scenario, essentially. Another area that I want to highlight is we're not only screening for one or two drugs, but we can actually do combinatorial screening, as shown here. So different small molecules used at the same time. So coming back to my previous image, so essentially taking iPS cells to all the way through somatic cells, we can do quantitative high-throughput screening at each step and optimize differentiation. And this, at the same time, entails using genomics, proteomics, metabolomics, and other analytical methods to really precisely understand and characterize cells. So back to the question of scale-up and standardization. So one approach that we're using currently is to actually use automated robotic systems. So in this case, we are using a system I want to introduce to you here in the next slide. So you have essentially this robotic system that can help us with dealing with the large amount of different cell lines that we are handling at the same time. So this will save, again, lots of time and effort and also standardize. And so we have a flask incubator on the left-hand side with hundreds of flasks. We can count these cells, we can test viability, we can do passaging. This is also image-based control here. And then on the right-hand side, you can actually plate these cells into plates that are compatible with for high throughput screening. So just to give you an idea of how this works, so quick overview of one of the steps during cell passaging. You see the flask is being opened here. So through those tubes, the flask will get media, and essentially a workflow that can be done 24 hours every week, so without any delays. And also making sure that we always change media at the exact time during the day, et cetera. So a great deal of helping us to standardize the technology. So this is another approach we're using. We're actually comparing different conditions side by side over protracted time periods. So in this case, comparing 4 different conditions, and this can be done over days, but also over many weeks. So making really sure that we are not stressing cells and selecting, but trying to uniformly differentiate these cells. Another area that I want to emphasize that is important to further introduce standardization is actually dose response. So using different factors, you have to make sure that you're actually stimulating cells and inducing genes. In this case, I'm showing an OLIG2 GFP reporter cell line. So making really sure that we are doing the right thing at the right time. Quality control, as I've mentioned, this can be further leveraged by actually identifying, characterizing novel surface markers. So Claudin-6 is a tight junction protein that is expressed by pluripotent cells, and it's downregulated when cells differentiate. Another area we are looking at carefully is systematically testing different culture conditions. So media components, cell density, different combinations and concentration of small molecules, et cetera. So really making sure that this is highly robust. One example, generating dopamine neurons, as I'm showing here. The next slide can make hepatocytes in 20 days, as I'm showing here, and this will be further used for a number of different assays, including toxicology, also for down the road for 3D bioprinting. This is an example where we have started making human skin actually using 3D bioprinting. And another area that I'm personally highly interested in is phosphoproteomics, an area that has been understudied so far in the field. So we can compare cells side by side. In this case, this study entails studying pluripotent cells compared directly to multipotent neural stem cells that we derive at high efficiency. So we can now look at complete pathways in a different way, and quantitatively, but also looking really at the big picture, not only just at 1 or 2 components of a pathway. So another example, how we mapped the epigenetic regulators in this study. So they are themselves regulated during cell differentiation. So transcription factors, very important area in this field, but it turned out that we don't have enough information on the protein level. So in this study, we are demonstrating that actually the vast majority of transcription factors are phosphorylated, and the study also identifies thousands of novel phosphorylation sites, and this includes even these extremely important transcription factors like OCT4, NANOC, and SOX2 being used for reprogramming, for instance. Another area that I wanted to highlight here is actually there are many other proteins and transcription factors out there that we actually haven't even thought about. So essentially, this study enables us to look at what transcription factors are expressed in other proteins only in embryonic stem cells or iPS cells, or then further differentiated cell types. And you see here comparing embryonic stem cells in this case and neural stem cells side by side and quantitatively how this dynamically changes. This is an experiment done in 6 days. Another story I wanted to briefly highlight is using such an approach, it's possible to actually identify novel mechanisms. In this case, we found that midkine is expressed by pluripotent cells and strongly upregulated actually when cells decide to differentiate. And so this turned out to be actually very important in the process of neural lineage entry. So I'm showing here that an autocrine paracrine mechanism is responsible for neural lineage entry. And if we block this feed-forward upregulation of midkine, we essentially disrupt neuralization. And I can't go into all the details, but please take a look at the published paper for more details. So finally, we have put this together as a new searchable resource. I'm showing here. You can also find this on the internet, and actually, this is currently the most expansive proteomics resource. So we have identified 13,000 proteins and almost 60,000 non-redundant phosphorylation sites. So coming back to the question of quality control, so I think we have to come to a consensus how we can use and derive information from all these different areas that I've highlighted here so that we get a good idea of cell characterization, so making sure that we precisely characterize cells, but also make sure that this will help us in the next step of translating this technology into the clinical scenario. So the goal is at NIH to come up with a iPSC portal. So this is a new website that will be launched. So we want to use this as a collaborative reference center and provide information on the basic biology of iPS cells, but also make sure that everything we are investigating and protocols that we are developing and the quality control standards are being shared with the public, including demonstrating translational utility of this technology. Okay. Finally, I want to thank all my colleagues at NCATS and also the NIH Common Fund supporting this effort. And finally, want to thank you for your attention.